Hello students, today we'll discuss about immunoelectrophoresis. The term immunoelectrophoresis was first coined by Graver and Williams in 1953. It is a process or a technique in which an antigen mixture is first separated into its component parts by means of electrophoresis, and the presence of the antigen of interest is detected by the formation of precipitin arc or line by immunodiffusion in agar. Principle: electrophoresis is the movement of macromolecules in an electric field, and migration of molecules depends on charge and size. When electric field is applied to a slight layer with gel, antigen mixture placed in vials is separated into individual antigen components according to their charge and size. Following electrophoresis, the separated antigen are reacted with specific antisera or antibody placed in trough parallel to electrophoretic migration, where diffusion is allowed to occur. Antiserum present in trough moves toward antigen components, resulting in formation of separate precipitin lines, each indicating reaction between individual proteins with its antibody. The procedure: immunoelectrophoresis involves two distinct and separate steps. In the first step, suitable holes are punched in a buffer agar plate. The holes on the agar plate are filled with antigen, and the proteins of the antigen samples are separated lengthwise through the agar by electrophoresis. After completion of this step, a suitable trough is made in agar, parallel to the length of the antigen migration. The trough is then filled with antibody and allow it to diffuse. The antibodies diffuse laterally to meet diffusing antigen under control condition, and lattice formation and precipitation occur, permitting determination of the nature of the antigen. Precipitates are deposited as arc between the antibody trough and the antigen track. The buffer used in the gel have an influence on electrophoretic mobility and on the position of the immunoprecipitate. The pH of the buffer must be chosen with the isoelectric point of the antigen in mind. It is desirable usually to work at a pH slightly above the isoelectric point so that the proteins have a negative charge and do not interact with the agar. In immunoelectrophoresis of different sample, it is possible to evaluate the amount of an antigen semi-quantitatively by the measurement of the distance of a certain precipitation arc from the antibody trough. In another way, immunoelectrophoresis is also called gamma globulin electrophoresis or immunoglobulin electrophoresis method. It is also used in determining the blood levels of the three major immunoglobulin: immunoglobulin M, that is IgM, immunoglobulin G, that is IgG, and immunoglobulin A, that is IgA. Types of immunodiffusion to see the precipitin arc due to antigen-antibody interaction. First, single immunodiffusion. In single immunodiffusion, only the antigen will move and antibody is immobilized, and it can be of single diffusion in one or two dimensions. Number 2, double immunodiffusion. In double immunodiffusion, both the antigen and antibody move toward each other and form a precipitin lines. Double diffusion can be of one dimension or two dimensions. During the precipitin reaction, three major zones of the antibody and antigen concentrations are formed. They are: number 1, zone of antibody access, where antibody concentration is higher than that of the antigen. Number 2, zone of equivalence, where the concentration of antigen and antibody are same. Number 3, zone of antigen access, where antigen concentration is higher than that of the antibody. Precipitin lines will form at the zone of equivalence, where the concentration of antigen and antibody are same, and it is the optimum condition to form the antigen-antibody lattice. Types of immunoelectrophoresis. There are 4 types of immunoelectrophoresis. First, electroimmunoassay, also called rocket or Laurel rocket. Second, immunofixation electrophoresis. Third, immunoprecipitation of protein after capillary electrophoresis. Fourth, classical electroimmunoassay. So, in number 1, electroimmunoassay, which is also called as a rocket or Laurel rocket. Electroimmunoassay or rocket immunoelectrophoresis is a simple, quick and reproducible method for determining the concentration of a specific protein in a protein mixture. To obtain an accurate concentration, the proteins in the reference solutions and in the unknown samples have to be physicochemically and immunologically identical under investigation. The samples to be compared are applied in well punched out of an agarose gel containing the corresponding monospecific antibody. One-dimensional electrophoresis is performed where interaction between antigen and antibody takes place until a point of equivalence is reached. The end result is rocket-shaped precipitation formation. Then, by measuring the height of the precipitate peak, the exact concentration of protein can be determined. Number 2, immunofixation electrophoresis. Immunofixation electrophoresis is a technique for detecting and typing the monoclonal antibody or immunoglobulins, that is IG, in serum or urine. It is of great importance for the diagnosis and monitoring of certain blood-related diseases such as myeloma. It takes place in 2 steps. In the first step, the IG content in the serum or urine are deposited on an agarose gel, followed by separation according to their electrophoretic mobility under the effect of an electric field. Once IG is separated, second step is the migration of serum proteins. Then specific anti-IG antibody are used for treatment by adding individually to each migration lane. If there is presence of monoclonal IG, appearance of a narrow band will be observed as precipitates. Number 3, immunoprecipitation of protein after capillary electrophoresis. The step for performing an immunoprecipitation can be done in 2 different ways. In one step, an antibody, which is a monoclonal or polyclonal, against a specific protein is pre-immobilized into an insoluble support like agarose or magnetic beads, which is followed by incubation with cell lysate containing the target protein. Gentle agitation of lysate during the time of the incubation period was done to allow the target antigen to bind to the immobilized antibody. Then the immobilized immune complex are then collected from the lysate, eluted from the support and analyzed based on the nature of the target antigen. In another, free unbound antibody is allowed to form immune complexes in the lysate, and the complexes are then retrieved from the beads. Lastly, number 4, classical electroimmunoassay. The analysis of Ed-Modem-Gerber is the classical method of electroimmunoassay which depends on antigen-antibody interaction. Here, the antigen protein are separated first by means of electrophoresis. Then, antibody is applied in a trough just next to the separated antigen of protein. Immunoprecipitates are formed after allowing diffusion for a period of time with the separated protein and antibody against each other. The use of this assay has given great impact in the protein chemistry. The important observation made were the great number of different proteins in serum, the existence of several immunoglobulin classes, and their electrophoretic heterogeneity. Results and interpretations. First, presence of elliptical precipitin arcs represents antigen-antibody interaction. That is, if the antibody is homogeneous, only one precipitin arc observed and multiple in case of heterogeneity. 2. Absence of formation of precipitate suggests no reaction. 3. Different antigens, that is the protein, can be identified based on the intensity, shape, and position of the precipitation lines. Applications of immunoelectrophoresis. First, the test helps in identification and approximate quantization of various proteins present in the serum and created a breakthrough in protein identification and in immunology. Second, it is used in patients with suspected monoclonal and polyclonal gammopathies. 3. The method is used to detect normal as well as abnormal proteins such as myeloma proteins in human serum. 4. It's used to analyze complex protein mixtures containing different antigens. 5. Used in medical diagnostic where certain proteins are suspected of being absent. For example, hypoglycemia or overproduce. Example like multiple myeloma. 6. They are useful to monitor antigen and antigen-antibody purity and to identify a single antigen in a mixture of antigens. 7. Immunoelectrophoresis is an older method for qualitative analysis of M proteins in serum and urine. 8. This technique aids in the diagnosis and evaluation of therapeutic response in many disease states affecting the immune system. The limitations of immunoelectrophoresis. First, immunoelectrophoresis is slower, less sensitive, and more difficult to interpret than immunofixation electrophoresis. Second, this method fails to detect small amount of monoclonal M proteins because the most rapidly migrating immunoglobulins present in the highest concentrations may obscure the presence of small M proteins. 3. They are limited in food analysis due to availability of specific antibodies. So in conclusion, immunoelectrophoresis is a powerful analytical technique with high resolving power as it combines suppression of antigen by electrophoresis with immunodiffusion against an antiserum. The interpretation of results depend on the formation of precipitin arc or lines between the reacting antigen and antibody. There are different types of immunoelectrophoresis with different applications. The main achievement of immunoelectrophoresis is that it has been able to identify a number of antigens present in serum. It has now been used in various medical, diagnostic, and research purposes.